Thank you. I want to thank the organizers for inviting me to speak in this session. I'm going to talk about deep genomics. This is my perspective. I'll show you how I see this world in 5 short slides. Then I'm going to talk about 5 studies from our labs, what we do with this deep genomics, and then I'm going to discuss how we're trying to deal with the ethical aspects of our studies. So fact number 1, there's DNA everywhere. This is an invisible world. We can't see it. Now I'm scattering DNA around me. And when I eat lunch, I eat DNA in my hamburger and in my sushi. The previous speaker also left her DNA around here. But we can't see it. And with advanced technologies, this invisible world is becoming visible. We can see it, and we'll see it in our databases in the coming years. Fact number 2, this is how our DNA looks like. This is a genetic code, A, G, T, and C. We're trying to understand it, and we're getting much better at it. We could recognize genes, we could recognize regulatory sequences in the DNA, and the larger the database gets, the better we're getting in understanding our DNA code. Fact number 3, in 2001, when the first human DNA was sequenced from beginning to end, it cost the equivalent of 4 jumbo jet airplanes, $1 billion. Now, with American incentives, we have better machines now that read the DNA much faster and much cheaper. So today the cost is a flight to the USA from Israel. That's the cost of sequencing your DNA. We know what the cost would be in a few years. It would be roughly an EasyJet flight from Israel to Europe. So DNA is everywhere. It's easier to read it. It's cheaper to read it. And we're going to have a lot of it soon. Now, the thing is, these machines could read DNA, but they can also read RNA. So we could look at genetic and epigenetic. We could fool the machines. We could squeeze out all the RNAs from our cell. We could use an enzyme that turns it into a DNA. And then we could read the RNA as well, or the RNA equivalent on these machines. So we could read everything in our cells, all the DNA, all the RNA, genetic and epigenetic regulation. We could also read the proteins on other machines. Not only that, we can also read the microbiome. So I am myself and all the microorganisms that I carry with me. A few years ago, a very nice study from the US, published in PNAS, showed that you can identify an individual based on their microbiome. So if you just take the sum of all the microorganisms in one person, you can identify them in the room if you go and then sample different items in that room. And fact number 5, we want to read everybody. Not only we want to read everybody's DNA, RNA, microbiome, we also want to look at their clinical data. We want to understand if someone suffers from a disease. We want to compare their DNA to the rest of the population and understand whether it's a genetic change, or maybe it's an epigenetic change, maybe it's an RNA modification that we could recognize, we can understand why or how this disease has developed. We could use this maybe for diagnosis and eventually for therapy. These are 5 facts. This is how I see the genomic world. Now, 5 studies from our lab. 1 slide on each study. Schizophrenia is a devastating mental disorder. It's genetic based, so if you have identical twins and one of them has schizophrenia, there's about 50% chance of the other twin to have schizophrenia as well. That means it's genetic, but also epigenetic. So we took nasal biopsies from the upper nasal cavity, 3 from the left nostril, 3 from the right nostril. This is the closest you could get to the brain without a major operation. We took these cells, we grew them in a plate. When the plate was full, we took out the DNA and also the RNA. We put the DNA aside. We profiled 20,000 mRNA molecules, 2,000 microRNA molecules, all the RNAs in the cells. And we wanted to see whether there's a difference between 20 schizophrenia patients and 20 controls. We found 1 RNA molecule there that you could see that is elevated in the live schizophrenia patients versus controls. So first of all, we see that schizophrenia might also be associated with epigenetic changes. What we're saying here is that if we profile RNAs of a much larger cohort, maybe we can assist in identifying schizophrenia patients not only by psychiatric evaluations, but also molecularly. In another study, we looked at trying to fit the right drug to the right person at the right dose in major depressive disorder. So we used 80 blood cells, 80 cell lines, lymphoblastoid cell lines. We exposed them to antidepressive drugs. We took the ones that really suffered and the ones that grew very rapidly. Instead of looking at the DNA, we profiled the RNAs there. We found one RNA highly elevated in the sensitive cells versus the non-sensitive cells. This RNA is involved in neurogenesis, or connectivity between the different neurons. We're investigating it further, and we started a very large cohort of patients. We have collected up to now about 60 patients in their first admittance to hospital, and then 1 month later. And we're going to profile the RNAs in these patients and understand whether what we found in our cell lines could also be applicable to finding the right drug in these patients when we're trying to compare responders to non-responders to drugs. We ran a study on Alzheimer diseased mice. It is known for many years that enriched environment, if a patient, Alzheimer disease patient, is exposed to an enriched environment, it could be physical, it could be mental enrichment, you can postpone the appearance of the disease. So we took Alzheimer diseased mice and placed them in enriched environment in cages with different toys, where you change the toys every week, and also you give them exercise, and other ones as controls in other very simple cages. A few weeks later, we sacrificed the mice, removed different parts of the brain, and we profiled the RNAs in different regions of the brain. What we wanted to see is the elevated RNAs in particular sites in the brain, and the ones that were elevated in the enriched environment might be the ones that extend the lives of these mice or prevent the early appearance of Alzheimer plaques. So we identified several of these RNAs in these brain regions. What we're doing now is we are reintroducing them into the brains to see whether we can prevent the appearance of Alzheimer in these mice just based on reintroducing these RNAs that were elevated in the enriched environment. We're working on post-traumatic stress syndrome in IDF soldiers, in Israeli soldiers. We're taking 3 time points. The first time point is upon their recruitment. Now, fortunately and unfortunately in Israel, we have healthy 18-year-olds from the entire population coming to the army, and we know that a certain amount of them will have PTSD. We're going to combat units where all these soldiers are exposed to traumatic events. So the first time point is upon their recruitment. The second time point is 1 year after their training. The third time point is after exposure to traumatic events. We also run this project together with Professor Talma Hendler from Sourasky Medical Center. She maps the brain using fMRI. Every time these soldiers arrive to the clinic, we also receive a blood sample from them, before and after the fMRI at 3 time points. They're mapping the connectivity in different brain regions. What we do, we try to find an RNA molecule in the blood that could indicate potential traumatic events. After running the first cohort, we believe we have one RNA marker in the blood that indicates that this soldier would be potentially susceptible to PTSD, meaning that it would require early intervention based on a molecular test eventually that we will expand on a larger cohort. And in our 5th and final study, we're looking at pregnant women. We're following thousands of pregnant women during the pregnancy. But the point is here that we're starting very early. From week 10, we're taking blood samples and then following the pregnancy. About 1% of these women will suffer from preeclampsia, a devastating disease with an unknown cause, with a major risk for both the mother and the fetus. What we're doing at the end of a pregnancy, after collecting all this data, we went back to the 1% that had eventually preeclampsia, but we went back to week 10, much before any of these women knew that they will suffer from preeclampsia. So we took blood samples, 0.5 ml of plasma, from 40 potentially preeclamptic women and 40 controls. We profiled 20 million RNA molecules from these blood samples. We ran a machine learning algorithm, and we found 25 RNA molecules identified at week 10 of pregnancy that would predict that this woman is under risk for preeclampsia eventually. Now, the good thing about our marker is that there's a solution for it. If these women would take aspirin, and this is approved in Europe already, a low-dose aspirin daily, if they start taking it before week 16, it will prevent 80% of preeclampsia in these women. So this is approved in Europe, not in the US yet. And another study we're carrying is by taking the blood of these pregnant women at week 10 and running a deep algorithm that can identify the DNA molecules in the blood, we can potentially, and we've shown it in several families, identify point mutations in the embryos of these women at a very early stage at week 10. And in some cases, the sensitivity and specificity is extremely high. In other cases, we know that there's difficult regions to see these mutations. We're trying to bring this identification to an earlier week. We're trying to reach week 5, where we will be able to take enough blood to profile these DNA molecules in the blood of these women. And by running our algorithms, we will be able to understand whether this piece of DNA arrived from the embryo or the mother, and whether it's inherited, this mutation or change is inherited from the father or from the mother. So these are studies from our lab, and I have very talented students, and we want to see how accessible the genomic, your own genomic DNA is. I'm taking my DNA everywhere, right? But I can't ask questions on my DNA if I don't make it easy enough. So one student in the lab wrote a software called Gene Genome, and in short, we call it GeneG. GeneG is GeneG is a redhead in Hebrew. It's a mischievous redhead child with DNA glasses that can run on any machine you have in your pocket. And you can start querying your DNA. If you upload your DNA, you could start querying your DNA and asking your DNA questions. Now, we're not giving you any medical information. We're just connecting you to other databases. Okay, so if I have a TT there, how do I compare to other people in the population? And what does it mean based on known databases? And you know, a male and a female before conception can bump their phones and look for common mutations to understand if they have a mutation in a similar gene or at the same location. Of course, this is just a demo we're running in our lab. It runs very well in the lab. The legal department at Tel Aviv University told me not to dare to get it out there, but we're trying to understand what it means when you have your DNA and what questions you can ask eventually. So together with Dr. Oren from the engineering department, we're trying to understand how we could limit the access or the information, but still maintain continuous research. So we asked, can we have a sensitivity measure of the DNA? If we take all the genes in our DNA, 20,000 genes, we build a model, we test the sensitivity. So we had a few students working on it. We retrieved data from known databases you can see here. One of my favorite one is PharmGKB, which is pharmacogenomics. That means that if you have a gene and you have a mutation, you can refer to what it means in terms of taking drugs and metabolism of drugs. Obviously, we also have a lot of data from the NCBI as well. So we had this, so we set up these scores. We call them inferential gene exposure scores. And these are true to the exact moment where we access the database. Obviously, and surely there's a bias here and a skew to genes that are highly studied or the genes that are very commonly known to be associated with major diseases. Here you can see there's a skew to the right-hand side. And if we look at the genes on the right-hand side, or the top 100 genes, it's not surprising to see BRCA there, and P53 and APC and ATM, major genes that are also highly studied, but also ones that are known to have many genetic modifications in several diseases. And also there's a big bias here, obviously, to cancer-related diseases. So we took our IgE scores and we accessed data. This is data from PGP. PGP is the Personalized Genomic Project, the project which started about 12 years ago from Harvard, from Professor George Church's lab. And we used our scores, and we looked at the data there. The data includes clinical data and includes genetic data there. And we gave these scores and clustered the groups into two groups, those with high scores and low scores. people with cumulative mutations. This is the end of my time. I'll finish in a minute. Cumulative high IgE scores and lower IgE scores. And now we're playing with this data, and we're saying, what happens if I take out 50 genes, okay, 50 relevant genes? Will these clusters get closer? And indeed, they get closer. And now we're taking out 1,000 genes. We're taking out 1,000 genes, and we're seeing these clusters getting closer together. That means that I'm taking information out of my database. I'm taking the highly ranked scores. I don't have to take out 20,000 genes. I can take out about 1,000 genes, and these clusters get closer together. Now, obviously, I'm losing data here. I'm losing here genetic mutations and genetic information, but I am able to retrieve this data or bring this data closer and mask it a bit so researchers could look at it but not define this DNA or genome or individual as a diseased individual or potentially diseased individual. So since I'm working with the engineering department, right away they started also an app which they call augmented genomics. And over here, we're allowing the patient to decide at what threshold they allow the other party to look at. That means they could retrieve 100 genes out, 500 genes, or 1,000 genes out based on the threshold they decide to expose. So maybe if they sit in front of their physician, they'll give high access to their DNA. Maybe if they're sitting in front of their insurance, they'll have a very high threshold. So that means their DNA would be masked. There'll still be a lot of information there, but the person sitting in front of them will not see their high IgE scored genes. So this is my perspective on deep genomics analysis we run in our lab and how we're trying to deal with these ethical aspects when accessing this data. Thank you.